Hello everyone, so in this video we are going to introduce to you about hyaluronidase. As we all learned before about different kinds of enzymes applied to different aspects of our daily life like baking, winemaking, medicine and so on. Hyaluronidase is a type of enzyme that we all know enzymes are absolutely made out of proteins, and the amino acids of the protein that give the 3D structure of the enzyme itself. While hyaluronidase, it has a chemical composition that made of thousands of carbon, hydrogen and hundreds of nitrogen, oxygen and sulfur. It has a molecular weight of 53,871 and 0.08 gram per mole. Every enzyme has its own EC number. As well as hyaluronidase, it has an EC number of 3.1.2.35, in which the 3 represents the major class of hydrolase, 1 represents the glycosidase subclass, 2 represents the glycosidase subclass, and the 35 represents the serial number of the hyaluronidase itself. Well, this enzyme is initially discovered by Durabranos, who a microbiologist that defined it as a spreading factor from the Streptococcus strain of bacteria in 1929. Meanwhile, this enzyme could also present in human body as a role in cancer development in terms of facilitating the invasion of the tumor cell, and also in immunity it facilitates the white blood cell for its migration, and even in fertilization it facilitates sperm to penetrate the ovum. Meanwhile, this enzyme can also be found from wildlife animals, snake, honeybee, and even a lionfish, in which both of these animals possess a venom and it has the enzyme in them. So now we move on to the substrate, which is the hyaluronic acids. Hyaluronic acids, they are typically a long-chain polymer formed by an N-acetyl beta-D-glucosamine and the D-glucuronate residues, and they are found as a part of extracellular matrix. Hyaluronidase acts on the hyaluronic acid by random hydrolysis to cleave the 1,4-glycosidic bond on the polymer to forming the monomeric units. And this substrate can be largely found from the human eyes, from our synovial fluid of our cartilage, and even from the dermis layer of our skin. They have its role in the wound repair, in the granulation, in the cell migration, and even the skin healing, in which both of these steps are closely related to one another in the function of the
In the medical field, Vitrace is widely used. So now we are going to look at the application of Vitrace. Vitrace, also known as hyaluronidase injection. Vitrace is commonly supplied as a sterile, non-preserved and colorless solution with a pH of 6.4 to 7.2. The method to inject Vitrace into our body is through the subcutaneous injection method. In this type of injection, after the drug is drawn into the syringe, a short needle is used to inject the drug into the subcutaneous layer, which is between the skin and the muscle tissue. Next, what can Vitrace do? First, Vitrace can be used to increase the dispersion and absorption of other injected drugs or medication. Second, it is used together with fluid injected into the body to treat dehydration. Third, it can also be used to prepare the purified bovine testicular hyaluronidase. Lastly, hyaluronidase is also used to help contrast dyes in our body to show more clearly on certain types of X-rays or scans. The next application is the combinant treatments. Combinant treatments are used in drugs such as Hyaluronidase. Hyaluronidase is composed of immunoglobulin infusion 10% from human and recombinant human hyaluronidase. Its function is to treat primary immunodeficiency (PI) in adults. It is infused under the skin, which is a fatty subcutaneous and also the subcutaneous tissue. So why hyaluronidase is used in the treatment? So during injection, hyaluronidase is injected to the subcutaneous tissue. When injected, the hyaluronic acid in the body is broken down by the enzyme, and this may increase the permeability of the tissue to fluid. Thus, it will make it have a better absorption and increase its dispersion.
And now, we will discuss about the role of hyaluronidase in cancer. So, as we all know, the phospholipid bilayer serves as the cellular barrier. At the intracellular site, there is the presence of cytoskeleton, whereas at the extracellular site, there will be the extracellular matrix. And the substrate for the hyaluronidase, which is the hyaluronic acid, is the main component of the ECM. Increase in ECM is a malignant phenotype of cancer and tumor, and this will lead to the increase in the hyaluronic acid and further increase the interstitial fluid pressure. This increase in the interstitial fluid pressure will then squeeze the blood vessels and reduce the diameter of blood vessels, so that it will be hard for the anti-cancer drug to reach the site. Therefore, hyaluronidase can break down the hyaluronic acid to prevent this condition to happen. There is controversy present in the role of hyaluronidase in cancer due to contradictory observations. Hyaluronidase are said to be supporting tumor progression, as degradation of ECM surrounding the tumor cells help the cells to escape from primary tumor mass and lead to the spreading of cancer. Therefore, hyaluronidase inhibitors can be used to inhibit hyaluronidase activity.